My name is Arjun, and my review paper was on the emerging role of oligodendrocytes in Alzheimer's, and my mentor was Rahul Patel. So I want to first start off by looking at a kind of like zoomed in section of the brain. So right now we're looking at the occipital lobe, but this could apply to basically any region. But you can see that in this image, we have neurons, but then by themselves, neurons wouldn't allow the brain to function like completely. There are a lot of helper cells called glial cells that are needed for proper brain function. So we have oligodendrocytes, microglia, and astrocytes. But specifically, my paper is focusing on oligodendrocytes. So oligodendrocytes are these really large branch cells that sometimes have up to 60 arms that all connect to these neurons. And what they do is they're responsible for creating this fatty substance called myelin, which basically coats the axons of these neurons and it allows action potentials to travel faster down these axons. And myelin has been linked to cognitive processing. And because these oligodendrocytes are very vital for the proper functioning of neurons and overall health of the brain. And therefore, if something were to happen to these populations, you would observe altered brain behavior. And that's kind of my hypothesis for the involvement of oligodendrocytes in Alzheimer's. So specifically, let's first look at how myelination happens. So oligodendrocytes stem from a precursor cell, which I will call OPCs, also known as Ng2 plus glia. And then these cells then mature into oligodendrocyte precursors and then eventually become myelinating oligodendrocytes. So these OPCs originate in the subventricular zone of the brain and then eventually migrate to regions where they're needed. So for example, if there was some injury like that demyelinated the region, what would happen is these cells would migrate to that region, mature into these myelinating oligodendrocytes. And then what would happen is they bind to the axons and basically coat them with this myelin. So let's look at the pathology of Alzheimer's. So beginning with a healthy brain, you can see here on the left that because these axons are really long, they have these microtubules. And what happens is along these microtubules, there's this protein called tau, which binds to them and stabilizes these microtubules. So it's called a microtubule associated protein. There's also a protein called amyloid beta, which is generally responsible for the homeostasis of neurons. What happens in Alzheimer's, however, is these amyloid beta proteins misfold, and they form these thermodynamically stable aggregates called amyloid beta plaques. And what also happens is these tau proteins that were bound to these microtubules, they get hyperphosphorylated, which means that a lot of phosphate groups are attached to these proteins. And when that happens, they basically fall off these microtubules. And what happens then is these microtubules disintegrate, and therefore the axons of the neurons also disintegrate. And because axons are really important for allowing action potentials and neurotransmitters to travel down from the soma or body of the neuron all the way down to the synapses, what happens is when these axons get disrupted, neural transmission gets disrupted, and eventually it leads to neurodegeneration. So if you look at the MRI image here on the left, that's an image of a healthy brain. But if you look at the image here on the right, you can see that there's a lot more black space. And what that's indicative of is that there's a lot less brain matter in Alzheimer's. And that's because of the neurodegeneration. Also, you can see that over time, the axons kind of disintegrate, as you can see in the image. And these amyloid beta plaques inhibit the synapses and dendrites, which blocks neural signaling. So linking this back to oligodendrocytes. So normally under homeostatic conditions, these oligodendrocyte precursor cells, OPCs, are able to mature into these myelinating oligodendrocytes and normally myelinate the axons of these neurons. However, what happens as these amyloid beta aggregates develop and these tau neurofibrillary tangles develop, they inhibit this maturation into the myelinating oligodendrocytes. And so there is a reduced myelination of the axons. And if you think about it, this could lead to a lot of issues because not only does this myelin allow for faster signaling, it also protects the axons of these neurons. So there are many different lines of evidence that implicate oligodendrocytes in Alzheimer's. So let's look at a couple of them. So in mice models, there's many different genetic models used to investigate Alzheimer's, but specifically I researched 3xTG, 5xFAD, and APP-PS1. So I researched what the current research in these mice models are, and I've summarized them in this table. So beginning with 3xTG, there was a notable decrease in OPC proliferation. So they're not dividing as much. They're not forming as many myelinating oligodendrocytes. There was also oxidative stress and elevated apoptosis that's cell death of these oligodendrocytes. In the 5X FAD model, there was an altered OPC transcriptional profile, which suggests that in a healthy brain compared to Alzheimer's, these OPCs are kind of doing different things, which suggests that they're playing some part in this pathology. Then in the APPPS1 model, there was a decrease in OPC density. So there's not as much of a population of these potentially myelinating oligodendrocytes. And there was also increased sensitivity to amyloid beta toxicity. And this led to calcium dyshomeostasis and also increased production of amyloid beta. And then finally, in vitro, so in a Petri dish, amyloid beta basically induced oligodendrocyte aging via upregulation of certain cell cycle control factors. It also, a specific subpopulation of oligodendrocytes called plexin B3+ OPCs were actually found to secrete amyloid beta. So instead of just playing a passive role in this pathology, that one study found that they were actually actively secreting amyloid beta. So my review paper basically proposed this cyclical model for amyloid beta and myelin in the brain. So myelin has a really high iron concentration, and that's because the reason for this is iron's an important cofactor in enzymes responsible for producing myelin. But what happens is with aging, and as this myelin starts to break down, iron actually encourages the fibrillization of normal amyloid beta into those toxic aggregates. And so once those toxic aggregates form, they cause further myelin breakdown and dysfunction of OPCs. And so there's this kind of positive feedback loop happening here where myelin breakdown causes amyloid beta production and amyloid beta causes myelin breakdown, which kind of contributes to the pathology of Alzheimer's. Also looking at MRI studies, which were specifically looking at myelin. This specific paper did it using a new technique called Q-space myelin mapping. I don't want to go too much into that, but it just allows you to look at myelin in the brain. But you can see that the yellow regions, the first row is in a healthy brain and the second row is in an Alzheimer's brain. You can see that there's a clear difference in the yellow regions, which indicate myelin. And what they actually found was that there was a decreased density of myelin with aging and also with Alzheimer's in important regions of the brain, such as the cingulate gyrus and the hippocampus. Another interesting discovery was that with there's a protein in myelin called myelin basic protein. And some studies found that this myelin basic protein was actually able to decrease the amount of amyloid beta present. So if you look at the image here on the left, the green and like the brightness is a fluorescent dye that binds to amyloid beta. And what they saw was that with myelin basic protein, amyloid beta concentration was actually reduced. And what this means is that myelin normally confers stability or a protection against amyloid beta aggregation. However, with myelin breakdown, oligodendrocyte dysfunction in aging, there's probably some role that oligodendrocytes play where this amyloid beta is allowed to start aggregating. And then finally, with tau in wild-type mice, it was noted that oligodendrocytes were actually taking tau from certain parts of the brain and spreading them to other regions, so kind of propagating this pathology. Also, it found that higher myelin amounts protect against tau pathology, which makes sense because they're protecting the axons of the neurons. So these tau molecules bound to these microtubules can't really get hyperphosphorylated. And so they're able to still tightly bind those microtubules. Then lastly, in humans, MRI and PET imaging indicates that there was decreased tau uptake in myelinating cortical regions. And so ultimately, my paper proposes this model where amyloid beta and tau cause demyelination of axons, inhibited differentiation of OPCs, and reduced proliferation of these precursor cells. And these are my references.
Great job, Arjun. Let me just rearrange things here. Great, we have a question from the chat. Does the lack of iron in myelin contribute to brain tumors?
I'm not aware about the role of iron in forming brain tumors, but just oligodendrocytes and myelin have the highest iron concentration amongst other cell types in the brain. And so that iron concentration plays a role in amyloid beta aggregation.
Great. From Jen, are you thinking about taking this research further in college? And has this experience informed what you might want to study in the future.
Yeah, definitely. I'm actually hoping to do some research this summer, probably some in vitro experiments with amyloid beta and oligodendrocytes.
Awesome. Seems like a great next step for the work you're interested in. Jonathan said, do you know anything about the recent controversy bringing into question the role of amyloid plaques?
Yeah, definitely. And this was something I think I forgot to mention earlier, but amyloid beta is kind of, it was initially thought to be the main key player in Alzheimer's pathology, but we're now learning that tau is playing a lot of a bigger role. And so my understanding of it from my research was still that amyloid beta is playing a role, but it's instead causing further elements like tau pathology later down the line. And so I was trying to incorporate these oligodendrocytes into a model that affected both amyloid beta and tau. So yeah.
Great job. Thanks so much, Arjun.